All right, so we're back with what should be our fifth and final section on energy and enzymes. And most of what we're going to deal with in this section is kind of a recap on basically the whole chapter, but also a description of how enzymes activity can be controlled. So, one very common thing that we see in cells is the presence of what are called metabolic pathways. So, we mentioned this before, I think, but typically for some product to be made inside of a cell, normally it's not made just in one step. It's made after a series of maybe several steps, maybe half a dozen or a dozen steps that eventually lead us from some initial reactant or substrate to some final eventual end product. And that's, like I said, a very common thing to see in cells functioning. So one of the advantage of this is that these enzymes, when they're part of the metabolic pathway, can be located relatively close together. They can be controlled as a single functional unit, and each step can also be controlled independently. Right, so that's a very common situation that we'll see here as we go along. And so a lot of processes that we see in living organisms, like blood clotting, for example, are the result of metabolic pathways, multiple steps, multiple enzymes that eventually give us some kind of final end product. Each step requires a different enzyme. Each step can also be controlled. So eventually we have this final end product that's produced. Now, in terms of control of enzymes, there are a couple different types. There are competitive inhibitors and there are non-competitive inhibitors. And that second one, the non-competitive, also goes by the name of allosteric inhibitor. So we'll talk about how those both work. Now, a competitive inhibitor is some kind of a substance, some kind of a chemical that bonds with the enzyme's active site and therefore prevents the correct substrate from fitting in there and going through a reaction. So it's kind of like a false key that blocks the correct site on the lock so that the actual key can't fit into the lock anymore. And so typically this is a kind of a reversible thing where you can have a competitive inhibitor come in, block the active site for a while when needed, when the enzyme needs to be shut off, in other words, and then eventually you can take it away and you can then start up the reaction again. There are some cases where competitive inhibitors are permanent, and in many cases those can be attributed to certain types of toxins or poisons that kind of forever block the activity of enzymes once they're present. So that's kind of a different story, but it is something which can happen as well. The other type of inhibition is called non-competitive, or like I said before, allosteric inhibition, where you have some type of a chemical that bonds to some other site on the enzyme that's not at the active site. It therefore changes the shape of the enzyme and changes the shape of the active site kind of by default so that the reaction can't occur anymore. So sort of think about, enzymes, again, are proteins. Proteins are super sensitive to anything that might change their shape. Well, if you bring a chemical along and bond it to the outside of an enzyme, that's really going to change its shape. And if the shape of the whole enzyme changes, the shape of the enzyme's active site will also change so that substrates can't fit in and go through reactions anymore. So that's another type of inhibition. You bond to the enzyme, it changes shape, and therefore the substrate can't fit anymore. So this is kind of the idea with a competitive inhibitor. We have the correct substrate, we have the enzyme, we have the active site, but in this case we have a competitive inhibitor bound to the enzyme so that the substrate can't fit in, and so the reaction can't take place. This is a non-competitive inhibition situation where we have another molecule bound to the allosteric site on the enzyme, nothing on the active site, but the allosteric site has now been bound. So the active site kind of changes shape by default, and now again, the substrate can't fit into the enzyme, and the reaction can't occur because the active site has changed shape. So, like I said, a lot of poisons, drugs, toxins, those kinds of things can sometimes bond to enzymes' active sites and prevent the correct substrates from fitting there. That will then therefore create the effects of the drug or toxin or poison by preventing reactions from occurring inside of cells. And as we said before too, some of these inhibitors are permanently bound, which can create some very serious problems down the road if someone has been exposed to these substances. We're going to kind of skip through this slide. Allosteric ones, again, we've talked about before. These are the non-competitive inhibitors, which can activate or deactivate enzymes. There are some allosteric molecules that will inhibit enzyme activity, and there are some that will actually increase enzyme activity as well. There are allosteric activators also, which are kind of like chemical on switches to activate enzymes and get them to function once something has been bound to that allosteric site. So there's actually both sides of the equation here. You've got an inhibitor and an activator when it comes to allosteric type molecules. Just a couple more pictures here to kind of drive this point home. We have a substrate, we have an active site in the enzyme. If this fits in, we eventually get some kind of end product as long as the reaction occurs. If we have an allosteric, in this case inhibitor, the enzyme shape has been changed, the active site's also been changed, and now the substrate can't fit in and the reaction doesn't work anymore. Anabolic reactions, a term you might see actually fairly frequently in some of your later courses. Anabolic reactions are any sort of reaction that is, first off, endergonic, but secondly, used to build things inside of a cell or inside of an organism. So if you're taking amino acids and hooking those together to form proteins, that would be a series of anabolic reactions. Because you're building complex things, this is also something that's going to be endergonic. It's, of course, going to require energy. Because as we said back at the beginning of the chapter, the universe doesn't like complexity, and so if you want to build something complex, you've got to be prepared to spend some energy to do it. Catabolic reactions, on the other hand, are those that break things down. When you take that protein and digest it, release those amino acids back into their environments, you're going to get some energy back from that as well. That would be a catabolic reaction because we're breaking down something complex into something simpler, and therefore energy is going to be released. That actually would be an exergonic type reaction. We talked about our activation energy. The little bit of spark that you need to get a reaction going in the first place is the activation energy. And we also talked about the relationship between the amount of activation energy needed and the rate of reaction. Enzymes then, of course, function by reducing the activation energy and also, therefore, speeding up chemical reactions. So just kind of like a quick review here, we have our illustration here of the breakdown of lactose into glucose and galactose. This guy here in this ball sort of represents the initial input of activation energy. Once you get to here, the ball, or actually in like literal terms, the reaction goes to completion and you actually get your two substances back in this catabolic reaction. If you have lactase present, now all of a sudden the activation energy is quite a bit less, and getting the ball to the top of the hill is a much easier task, and then getting it back to the bottom is much quicker as well. So again, notice the endpoint's the same. The ball's at the bottom of the hill, the same hill in fact, but this initial kind of bump that you have to overcome is much smaller when you have the enzyme versus when you don't. All right, so just kind of recapping here a little bit, enzymes are bound to substrates at the active site. This is a very, very specific complementary fit like a lock and a key. When that occurs, the enzyme pre-stresses the bonds in the substrate, making them more easy to break. That's what essentially lowers the activation energy, and that's what makes the reaction actually take place quicker. We have many, many ways of controlling enzyme activity. We could do it at the genetic level by activating or inactivating certain genes, thereby allowing genes to be expressed or not. We have allosteric activators that sometimes need to be sort of turned on to make enzymes function. We have various types of allosteric inhibitors as well as competitive inhibitors. We also have feedback inhibition. Kind of the last concept here to cover in this chapter is the idea of feedback inhibition, and we talked about this back in the last section where as products continue to build up, they can actually interfere with further reaction and therefore slow down the production of more products, which is kind of handy sometimes. So you don't waste essentially ingredients by making more products that you don't really need. Feedback inhibition helps you prevent that from occurring. So this is the idea here. We have threonine, which is an essential amino acid that we get in our diet, and we can actually convert threonine into isoleucine through a series of chemical reactions. The idea here is that as isoleucine builds up, as we have this amino acid in larger and larger quantities inside of our cells, this amino acid itself begins to slow down this first reaction and prevent threonine from becoming more isoleucine in the end. So once you have enough, don't make more. It doesn't make sense. It's a waste of energy. It's a waste of ingredients. So the buildup of isoleucine prohibits more isoleucine from forming. That is a really good example of feedback inhibition. So it's a nice kind of handy automatic way to shut down these reactions so that we're not wasting materials and so we're not wasting energy. So that's the end of our energy and enzymes chapter. From here we'll be moving on to cellular respiration, taking a look at how glucose is actually used in a cell to make ATP. And so we'll pick up with that section here next.